This is Biochemistry Practice Test 3, and it covers chapters 13 through 16. You will notice that some of the question numbers don't match. So in the sound part of the video, you will notice that I'm referring to different question number than one that is actually appearing in print and is actual question number. That's because I had to adjust a little bit this video from previous videos, remove unnecessary parts, and some of the question numbers do not match. But still it's going to be clear which question I'm talking about, and it would have taken too much time to re-record everything. So I apologize for that small inaccuracy, but it should not be a big inconvenience. Which segment of sodium channel is a voltage sensor and regulates opening of the channel? Well, there are 6 segments and it's segment S4. So segment 4 or S4. Next is simple nomenclature question. What is the name of the following compound? So you can see that base is adenine, and the sugar part is deoxyribose. So that's 2-deoxyadenosine. And there are 2 phosphate groups, so it's 2', actually not 2-deoxy, it's 2' because on carbohydrate it's 2' position, that is deoxy position. So it's 2' deoxyadenosine diphosphate. Question number 5. In the course of protein synthesis, which site on the ribosome can never be empty? There is only one out of the 3 sites. Those are A, P, and E sites, and only P site can never be empty. P or peptide site. Question 9. So question 9 refers to this illustration right here. It shows phosphate translocase. And so what is phosphate translocase? Is it an antiporter with H plus being primary transporter? Well, you can see on the right that H3PO4 minus and H plus are moving in the same direction. Antiporter means that they're moving in opposite directions. So that's not the case. B uniporter, that's only one species going one way or the other. So C, symporter with H plus being primary transporter, that looks right. That looks right because it shows low and high concentration. H plus goes from high concentration, you see it says higher H plus, to matrix lower H plus, so it's primary transporter, and then H3PO4 minus would be a secondary transport. And D obviously then is not the case. So C, 9C. 10. Note movement of ATP and ADP. So they are the middle one in black shown. How are they moving? So you can see that ADP is moving from cytosol to matrix and ATP opposite, from matrix to cytosol. So channel that allows their transport is obviously antiporter because 2 species are moving in opposite directions. Question number 11. ABC transporter transports ATP between the cells. That's not the case. Maintains HP gradient? No. Imports small molecules into the cell? That's not the case. You should look for the best answer. In some prokaryotes that could happen, but that's not general. General activity of ABC transporter is to export small molecule out of the cell. So that's correct. D. Always you have to pick the best answer. 12. Which of the following cannot be transported through gap junctions? So rule of thumb is mass of 1000 or 1 kilodalton. So if mass is 1000 or larger, it cannot pass through gap junction. If it's smaller than 1000, it can. Glucose, small, yes. Glycerol, small, yes. Transport RNA, no. Transport RNA or transfer RNA is pretty large. It cannot pass through. D, calcium, pretty small. Of course, it can pass through. So C, transfer RNA. 13. Which of the following is a second messenger? We just covered that. So CAMP is second messenger. Epinephrine is primary messenger. Protein kinase A is an effector. So second messenger will affect activity of protein kinase A, and then that one will cause actual physiological response. Sodium doesn't do anything. So 13A, camp. Question number 14. Upon activation by GPCR, which unit dissociates from G protein? So it's beta-gamma complex. Pretty much you have to memorize that. There is, in question like this, see, there is nothing to understand, and there is nothing to reason out. You cannot reason this out. Some of the previous questions, if you know the material, you have at least some basic knowledge, you can reason it out, you can figure it out. This one, questions like this, you have to memorize. There is no way around it. So 14C. 15. Which of the following pairs up with adenine, and how many hydrogen bonds are formed? So again, you may remember formulas, or you may memorize it like this. Correct answer is D, uracil, 2 bonds. Thymine also pairs up, but that's why I put 3 bonds, so that only one is correct. So be very careful about that. So you have to know both how many hydrogen bonds are formed and what is correct base, what's correct base pair. So it's not a trick question. I don't consider that to be a trick question because you need to know both. And you can see A and B. So what pairs up with guanine? Cytosine, cytosine A and B. But correct answer is A, 3 bonds. 16. In the molecule shown below, substituents X and Y are in which positions? So if you look at first X is in position 5. And actually that's not very best written question because C has X in position 5. So all you need to know, for the actual test, if I give you this, probably there will be 2 answers with 5 rather than just 1. So that you have to know both. So that already tells you that answer is C. So you have to know that on the base, regular numbers are used, 1 through 5 or 1 through 9. While on the carbohydrate part, ribose or deoxyribose, prime number. So Y is 2 prime. Question 17. Which of the following represents a nucleosome structure of DNA? So nucleosome is A. So again, those are something you have to memorize and recognize it. 18. D loop of tRNA has got its name. Again, another memorization question. Why do we call it D loop? Because it contains dihydrouridine, a rare base. So one of those unusual base, that's a better name. It's not really that rare, it's just unusual. It's not one of the 4 common bases. Question 19. This one is, unlike previous ones which were questions to memorize things, this one actually you need to understand the material. Which of the following would be a mutation that would not significantly affect structure and function of a protein? It's also called silent mutation. So on my exam or somebody else's exam, this question could be also, which of the following would be a silent mutation? Because it would be a mutation, but protein would still continue to function as normal, or pretty much as normal. Substitution of lysine with aspartate. No, lysine is positively charged. It's basic. Aspartate negatively charged side chain. Acidic. So no, that's really bad. That would be substitution of basic with acidic. That's bad. Substitution of serine with leucine. Serine is polar, leucine non-polar. Again, no. Serine capable of hydrogen bonding, leucine no, only Landau interaction. So that's also a big change. C. Substitution of valine with isoleucine. Isoleucine is maybe only slightly more hindered, more bulky compared to valine. But otherwise they are the same. They are both non-polar, capable only of hydrogen bonding. That would not change significantly the shape of the protein. That's silent mutation. That's one you're looking for. So C is correct answer. Just to check, let's look at the D. Substitution of glutamate with proline. First, glutamate and proline are very different. Proline forces U-turn, forces change in shape of the protein or protein chain. It's very rigid and it's non-polar. Glutamate is pretty polar. So no, definitely not. So C is correct answer. So 19C. So in the course of synthesis of hnRNA, that's actually a direct copy of DNA, direct transcript from DNA, the bond is formed by nucleophilic attack of the incoming nucleotide 3' OH on the polynucleotide's 5' phosphate, and the RNA molecule grows in 5 to 3 direction. That's actually correct. Then you have various variations. So you need to know how the chain grows. And all of those other variations, if there are two things, 3OH to 5' phosphate and 5 to 3, there are various variations, but correct answer is A. So in this case, first one is correct. On the test, I would advise you to read all 4, so to make sure that you eliminate wrong ones, that you're certain that you got correct one. 21. Which of the following involves hydrolysis of GTP to GDP? And that's actually synthesis of proteins, obviously. Synthesis of charge transfer RNA, dissociation of elongation factor from a ribosome, initiation of transcription, dissociation of transfer RNA from E site of ribosome. It's B, dissociation of EF from a ribosome, elongation factor from a ribosome. And question 22. Individual amino acids are attached to their corresponding transfer RNAs by forming an amide bond to 3' OH. No, they don't form amide bond. By forming an ester bond. That's the one. Ester bond to 3' OH in the acceptor strand. That's correct. And then you can check the other two. Forming phosphoester bond of 5'. No, that will be a very unstable bond. Wouldn't survive in aqueous solution. Forming amide bond. Again, amide bond is not formed. So 22B. So this completes the entire practice test. And at this point, you have all the material you need for the third test.